So my name is Mike Tarr. I'm in the psychology department and the Neuroscience Institute at Carnegie Mellon. I study human vision, but I've always tried to connect human vision back to artificial vision systems. And so this is a lecture sort of designed to contextualize the idea of how we can connect what we understand about the biology of intelligent systems with artificially intelligent systems, which is becoming something which I think is a lot more interest to people than it was maybe 10 years ago in various ways. So one of the things I like to start with is this hype cycle graph, which some of you have seen before. And I think it applies very nicely to AI. Applies to lots of other things, of course, too. But the idea is that you have some kind of technology trigger, something that creates a lot of excitement. In the case of recent AI developments, it was deep learning, which sort of came around in the early 2010 to 14 time period. Everyone gets really excited when that happens. There's always a lot of hype. If you read more popular press, in various ways people claim, oh, this is going to solve all of our problems. It's going to be really great. It's going to do amazing things. Then what always happens, you have this trial of disillusionment, and that means that all of the hype gets, damped down. People realize that it's a lot more complicated than they thought. And then what happens eventually is you begin to do real work over time, the hard work to making things really work, and you get to some point where things are really productive. And so I put this up here mostly for you to think about two things. One is, for AI and deep learning in general, where are we on this? People could argue, oh, we're still in the peak of inflated expectations. Someone could say, no, we're into disillusionment. If you read Gary Marcus, for any of you that bother to read anything by that guy or some of the other people that have been critical. Or maybe we're in the slope of enlightenment, we're getting to productivity in different ways. Another specific place which is fun to apply this is, of course, is autonomous vehicles. There was this period where everyone was going to have self-driving cars by, 2022, I think they said. And now, obviously, we're a little bit further away from that, but maybe we're figuring a lot of it out. So that's something to think about as we talk about this, and also in particular how we connect this back to biological vision. And by the way, people can interrupt me. It's fine. Can you hear me in the back, by the way? OK, good, because I'm not using the mic. So one of the things I like to think about when we talk about the connection between biological systems and artificial systems is, where are we in terms of a couple different dimensions? So the dimensions I think which are critical are, one is performance. And obviously these days, if you're interested in deep learning and AI, performance is a big metric. There's all these different contests, like the ImageNet competition. There's a lot of other ones that have been developed over time. And so performance is a big metric now for success. But of course, it's not the only metric for success, and we can think about other kinds of things that could be relevant. So one of them is biological plausibility. If you're interested in connecting artificial systems and biological systems, is how reasonable are the models you're building in terms of also explaining what's going on up here, as opposed to just performing really, really well. And those can be completely different metrics. You can have a model that performs terribly but does a lot of the kinds of things people do, or you can have a model that performs really well and explains a lot of things people do. And we're going to talk a bit about that specifically. OK. Another axis which I think of as slightly different from biological plausibility is cognitive plausibility. So that means at the level of algorithmically, how we think about problems, how we solve problems, just at a sort of meta-algorithmic level as opposed to the specific implementations, the kinds of units or computations are being used. Is there a similar style between what's happening in the reasoning in an AI system and a natural system? So one example of that might be, again, in autonomous vehicles. Actually, maybe some of you can answer this question. What's one of the biggest problems they're running into right now with building autonomous vehicles and making them work well? Anyone know? Anyone interested in that space? Thought about it at all? Read anything? So they perceive really well. At this point, they have LiDAR. They have all these cameras. They've got very good algorithms. They can perceive everything in the 3D layout of the world. They're actually really good at mostly object recognition, inferences, notwithstanding what happened in Arizona a couple years ago. They have really good layout of the whole scene. They can do it a lot of ways a lot better than a person because of the LiDAR. But they have a big, big problem. They have to deal with pedestrians, drivers, and bikers. People. People have funny different behaviors and social interactions. Like when you're trying to cross the street, let's say at Forbes and Craig, there's a lot of eye looking at the drivers and people deciding whether they're going to jaywalk or not and figuring out what's going to happen. Autonomous vehicles don't know how to do that. So we can imagine that we implement in the autonomous vehicle a whole set of social inferences about the way pedestrians are going to behave. And those could be cognitively plausible. They could be things that are reasonable in terms of the way we think about the world and the way we think about the way people behave that we want the car to be able to do as well, but they might have little to do with the way it actually works in our brains, and that would be completely fine. Okay, so there's these different levels at which things can correspond to biological systems or not. And I tried to put up here a couple different maps of or points about where we are with different kinds of technologies historically and currently. So there's a little bit of interesting history here I want to get into, partially because if you're at Carnegie Mellon, it's worth knowing a little bit about the history of AI. So Carnegie Mellon likes to claim we're the birthplace of AI. We're probably one of the birthplaces of AI, if we're a little bit more honest, along with probably Stanford and MIT, maybe a couple other places. But we certainly are one of the earliest universities to start talking about that, notably Herb Simon, who all of you have probably heard of, or if you haven't, you should go look him up on Wikipedia or something. In the 1950s, he and a lot of other people started to talk about AI systems, systems that thought, and what they mostly worried about wasn't really good performance at the time. It was really about explaining human psychology. And in fact, Herb Simon, for most of his career, was in the psychology department at Carnegie Mellon. And weirdly, for those of you that don't know this, he won his Nobel Prize in economics for his study of human behavior. So he was always concerned about AI as a tool for understanding how people behave. So he built systems, these symbolic systems. If some of you may be familiar with some of the old systems that they built, the most famous one probably is still around is ACT-R. Those systems are symbolic systems that are cognitively plausible. They reason about the world the way people reason about the world. Like notably, there was a lot of things that did a lot of diagnosis the way doctors do. They would take all your symptoms, they would take a bunch of your medical history, and they could reason about what was likely to be wrong with you. But they didn't have a lot of biological plausibility, and they actually didn't perform that well. But that was sort of early AI. That went through the complete hype cycle during the 1960s. My favorite paper that was an example of the hype cycle was it's MIT AI Lab memo number 1, and it was called the MIT Vision Project. And it was one page. You can download it from my webpage, actually. And it said, we're going to have a bunch of summer students this summer solve the vision problem. Okay, that was it, basically. Because they thought it was a fairly straightforward thing. You have a camera, you take the pixels in, you have some AI program that can read the pixels in, and you'll be able to interpret the whole world. So they thought vision problem was a summer undergraduate project. All right, so that's a little bit example of the hype. Then they went into this trough of disillusionment. And there was actually something during the late 70s, 80s, and early 90s called the AI winter, which AI was like the dark corner of all of computer science. It was considered like kind of embarrassing to be an AI researcher, and it wouldn't get funding, and everyone thought it was completely useless and kind of this funny stuff. Okay, obviously, times have changed. So that was early AI. Starting in the late 70s into the 80s, there was something called parallel distributed processing. Have any of you ever heard of that term? Okay, that was the early neural net literature. So neural nets became quite popular at that point. They tended to be single layer. They weren't multi-layer like we have now. They used backprop. That's when backprop was developed. Okay, so that's an important innovation. They did learn something. And everyone got very excited. There's these big volumes, these PDP volumes that actually came out partially from Carnegie Mellon. Jay McClellan in particular was a major driver for that. The PDP summer school was taught at Carnegie Mellon. There's actually a great picture I have of the first summer school. I forget when it was exactly, but it's got Jan LeCun and Jeff Hinton and all the fathers of modern AI in that photo. They were all here. So PDP was a major driver, but they didn't have some of the later technology or engineering innovations that sort of make deep networks work really well. So these things only worked marginally well. They showed catastrophic interference. They were slow to learn. They didn't generalize as well as they should have. But people were excited about them for a while. They performed a lot better than early AI in a lot of tasks because they learned in this kind of implicit way. They learned from data in a statistical means. They were a little bit more biologically plausible in the sense that they had lots and lots of small, simple units connected together, just like neurons in your head. They were not very cognitively plausible, which is why they were criticized so strongly and why they eventually sort of fell out of favor with most people. They seem to solve problems, but not in the ways people solve them. So that's over there. Then you've got in the 80s into 2000s, sort of that's really the AI winter for the most part. People kept trying to push performance forward in various ways, mostly through hacks and very special purpose sorts of things. They weren't real cognitively plausible. They weren't really biologically plausible. They were just trying to make systems that did something half decent. So particularly in computer vision, we learned a lot about geometry, built better computer vision systems. They weren't great, but they could do some tasks reasonably well. Like you could read circuit boards and diagnose whether the circuit boards were defective or worth producing as product automatically. There was other computer vision problems which you could do automatically. But it wasn't great. Face recognition was a really hard problem. So performance is mediocre. And then what happened, of course, really Jan LeCun was pushing in this in the late 90s, but he didn't have the computational power. It was really the advent of GPUs, along with these engineering innovations, particularly ReLUs and some of the other things that ended up happening that being implemented in the deep networks. And deep AI took off, and we've got to this point where suddenly performance shoots way up from everything before it. There is some biological plausibility, and in particular, you may not know this, deep networks were specifically inspired by the architecture of the primate visual system, the human and primate visual system. If you look at the visual system that you've all got, you've got a hierarchy of layers going from the earliest inputs, which are weirdly back here. They come in through your eye, they come to the back of your head. You start with these layers, and then you move through this progression of layers that are connected until you get to this higher level output. So it moves like this. Both Jan LeCun, Jeff Hinton, and Yashio Bengio also had worked in vision, knew about the primate visual system, and that was their inspiration for building these hierarchical neural networks. They were very much inspired by that. And the convolution process was also inspired by the architecture of your early visual system. It looks like it's doing something like a convolution. So there was this idea of some biological plausibility in deep AI. They'd given up on cognitive plausibility altogether. We're going to talk a little bit more about that, but there was no attempt to make it think the way people thought. It was really an attempt to get performance much higher and have it in some sense perceive the way people perceive. Any questions about that at this point? Anything anyone wants to ask? Okay, good. So, in the future what we'd like, I can't really do the 3D, but this is supposed to be the future, it's supposed to be highly biologically plausible, cognitively plausible, and even better performance. That's where we want to be, right? Okay, I want to make a little distinction between different kinds of AI because I think people confuse these things quite often. We talk about AI in different contexts. So there is some kind of AI I've alluded to already that maximizes performance. Sometimes you just want a system to perform a task really, really well, and you don't care how it gets there. And so it might be diagnosing a disease. Like if you're trying to diagnose skin cancer, and you wanted to build an app that photographed some mole you have on your arm, and then it came back and told you whether or not you were cancerous and you needed to have it removed or you were fine. You really don't care about if it works the way people think. You don't care if it works biologically the way your brain works. All you want to do is maximize how well it performs. That's your single goal, right? And there are a lot of cases in AI where you might want to do that. But there's also a lot of cases, particularly now as we move into sort of this current era, where that's not your ultimate goal. And we can talk about that. Okay, so then there's AI, which is meant to simulate or better understand cognitive or biological processes. So the early PDP, the parallel distributed processing, but even now, there are models which are really artificially intelligent models. They look like AI models, but they're really meant to correspond to understanding some kind of process in your brain or the way you think. And you're not caring about performance, really. Performance is your ultimate goal. Your goal is explanatory power. And if you think about it, it could be that the performance and the explanatory power diverge from one another because we're not perfect. And I'll have a demo at the end, a fun demo, if you hang through the whole lecture about why we're not, kinds of things why we're not perfect. So there is that kind of AI. You probably don't run into it so much over in SCS, but it is something that people do. And I think that models for understanding other sorts of systems are often reasonable. Then the kind of AI I'm going to focus on mostly here is AI that happens to perform well, high-performing systems, but also allow us to help understand cognitive or biological systems. And I think that's really where the sweet spot is that we're getting into right now, and I'll give you some examples of that. And hopefully there's some connection between performance and also explanatory power. Might not be again. And then the last kind of AI, which I think is important to think about, is AI that's meant to interact with humans. And so that means it might not perform at the ideal level, which it could. It also might mean that it thinks like humans at the cognitive level, but it doesn't necessarily have a biological correspondence to humans. But What you really need to do is predict human behavior and interact with humans productively. So Google, Netflix, autonomous cars, anything where it's interacting with us, it's an agent of some sort, what is it, Alexa, Siri, and so on. Basically, they need to have an understanding of the way we think and do what we expect them to do. So Alexa is useless if it does something different than we expect it to do every time we ask it to do something. That's just annoying, right? What you really want it to do is what you think you want it to do when you tell it something. So that's not perfect AI. It's high performing only at the level of our own metrics for what we want out of the world. And we know people aren't perfect and do stupid things all the time or ask stuff that isn't right. So you don't want it, for instance, you don't want it to be like your parents and say, like if you say, turn the volume up to some number, and then it would say, that's dangerous for your ears. You shouldn't listen to your music that loud. That's not what you want it to do, right? And there's a lot of cases like that. If it started talking about your nutrition when you're ordering food, like, why are you ordering from McDonald's? That's really bad for you. Here's some healthy choices you could choose. You would be really, really frustrated. You'd probably stop using it. But that might be more optimal in many ways. But that's not what we want out of these things. We want them to basically act like people. In the same way, the cars, they can drive better than people. But what we need them to do is just behave the way we expect them to behave as drivers. Otherwise, they're just annoying and frustrating. So, so one of the things that they've actually grappled with in autonomous cars, too, is when to break the speed limit. Because there's some places where people always break the speed limit, and there's a nominal speed limit. You probably all have experienced places like that. But everyone drives at another rate. And so if the autonomous vehicle is holding to the speed limit, everyone's just going to be annoyed and honking at it or trying to go around it, which creates a lot of other danger. So it has to behave the way people want it to behave, even though that's against the law or not optimal in some ways. So it's important to think about that kind of AI. I want to say a little bit more about deep learning. I'm sure you guys, since that's the topic of discussion, know most of this, but just highlight some particular things. So one of the breakthroughs, of course, that changed from the PDP models and most of the other AI till now was this idea that you could use really large-scale training sets. And of course, that was enabled by the internet, having things like Google, Facebook, having these massive data sets with millions of labeled inputs. That was a radical change. We just didn't have the scale or the computing power to deal with that kind of thing before. And it turned out that some of the kinds of neural net architectures we'd used in the past actually work really well when you give it enough training data. It was just a matter of training data spanning the space of what you need to learn well enough, and then you could learn it. Okay, obviously enough parameters to learn that space. One thing to remember, which I think gets lost a lot when people talk about deep networks in modern AI, is that labels are a metric of human performance. So the network will do as well as the quality of your labels, right? If you have crappy labels, if for whatever reason you had lots of dogs labeled as cats in your data set, it's going to learn the wrong mapping. There's no way for it to know what the correct mapping is. And I think people miss that a lot. We tend to use data sets that are labeled and just treat them in this kind of black box kind of way, but they really are just a metric of human performance where the network will never do better than what the labels actually provide. And so, for instance, if you think about modern vision systems or text systems that process information, the labels tend to be fairly sparse. So how many of you are familiar with COCO? Not a lot of you. It's common objects in context. It's sort of a better data set than ImageNet, which I'm sure all of you have heard of. ImageNet tends to be one object. COCO is multiple objects interacting, and there's multi-labels there. Well, you need that kind of multi-labels, but even for that, COCO is a little bit sparse because you have objects interacting, so there might be a kid kicking a ball and a soccer goal on the other side. And think about the different complexities of that, things you could label that as. It could be playing soccer, it could be kid, it could be ball, it could be soccer net, it could be kicking, it could be outside, it could be sport. The list goes on. So the complexity of the labeling really gets very detailed. And your own cognitive labeling of that, when you look at that picture, you apprehend all of that instantaneously. And labels tend to be relatively sparse compared to what we actually know about a scene. And I think the same is true for text often as well. So there's a real challenge in getting labels. So features might exist in the input that would improve performance, but unless they're sometimes correctly labeled, the model's never going to learn them. So models are only as good as the labels. I think that's a real challenge right now for a lot of AI, and this is why people have been moving to more unsupervised methods, where you can discover features, discover new categories, things without being bound to what the labels are. But for the most part right now, of course, people are still bound to the labels. Questions on any of that? Okay. So a network can also reduce misses, but it can't discover new mappings. I already made that point. in these correlations. Again, unless you have some kind of unsupervised learning that you build. And again, I think that's one of the challenges that we're trying to work on right now. And there's debates about how much people are unsupervised, and we can talk about that. Like as a baby, how much of the learning you do is with some kind of labeling or inputs versus being that you're discovering information about the world. Probably a little of both is the answer. So just to summarize, deep learning is very good at the kinds of AI that predicts human performance because that's the labels, right? They specifically are explicit about human performance. And you can be pretty good at maximizing performance, but it's not clear how well you can understand biological intelligence if you're just using labeled data. Because again, we apprehend everything about a scene. How do you study, and we understand, of course, that scene dynamically, if it's actually really in the real world, lots happening temporally as well. And most of the kinds of networks that try and explain that are all much sparse in terms of both the labeling and they rarely have the dynamics. Okay? And so you might also be able to use it as simulation for biological intelligence, but you have to understand the limits. And a lot of the critiques, by the way, if you read any of these things, like again, Gary Marcus is probably the most well-known person doing this kind of stuff. They're critiquing things which are kind of obvious if you think about these points. They're complaining about the fact that the models use labeled data, that they're sparse, and that they don't necessarily work in unsupervised ways. Okay, fine, but they don't need to complain. We know that. They don't need to complain as badly as they do. Okay, I want to ground this a little also in what's happening in our visual systems. So these are some numbers about the way your visual system works. This is a human visual system. Monkeys and apes are similar, but we're about as complicated as it gets. So your retina is the surface, the receptor surface in your eye, right? That's where the light comes in, and you have photoreceptors, which are light sensitive. There's actually a chemical called an opsin, which changes chemically when the photons hit it. You have about 10 to the 8th photoreceptors in each eye, all right? Your eye has 4 layers of processing in it. Before you ever get to your brain, you have 4 layers of processing. 2 which you can think of as vertical, it's transmitting information down or a vertical direction, meaning it's compressing in some way. And 2 which are horizontal, meaning that they're forcing interactions between local units so that they become more global in their scope in terms of passing information laterally. So it's a neural network in your retina, receives the information at the photoreceptor level, and then there's these processing layers right there in your eye. So your eye, I always like to think of as your brain pushed out into the world. Okay? The retina does less complicated processing, and it data reduces things so that you go from for every 100 samples you have in your eye at your photoreceptor layer, the optic nerve, there's only 1 sample. It's 100 to 1 data reduction. You go from about 100 million to about 1 million data points. So think about that. Right in your eye, you're reducing at that level. And sometimes people ask, is that a lossy compression or a non-lossy compression? And the answer is we don't know. But if I had to guess, it's clearly lossy. There's a lot of signal processing happening in your eye. Things are changing a lot. Okay, in intelligent ways. So my favorite what's going on in your eye story isn't actually about people, it's about frogs. So there's a famous paper by someone named Jerome Levitin. Anyone ever hear of him? He was at MIT, he was a professor in electrical engineering. He wrote a paper called What the Frog's Eye Tells the Frog's Brain. So in the 50s, he got involved in doing physiology, sticking electrodes into neurons and recording how the neurons are responding to different stimuli. Electrical engineers, that's what they would start doing, right, if they got interested in the brain because they were the ones that designed the electrodes. So he was recording from frogs' eyes, and he was looking at the ganglion cells, which is the layers below the retina after the photoreceptors, after processing's been done, but before it's gone into the brain of the frog. And what he found was that he had ganglion cells in the frog that were responsive only to dark spots of light in a specific location in space that moved. So what's a dark spot moving to a frog? Insect, right. It's a bug. It's a bug detector in the eye of the frog. Forget about the frog's brain. By the time the information is going to the frog's brain, it's already signal processed it down to the point where it's telling the frog there's something like a bug out there. So that's a really classic example of taking your data, turning it from an array of light values into something much more high level that's evolutionarily important to the animal. Okay? So he had this paper, Frog's Eye Tells the Frog's Brain. That paper was rejected from, I think, like 30-some journals. Everyone thought he was crazy. There must be some error. He finally got published in a weird, obscure electrical engineering journal, and it's one of the most cited papers, of course, of all time in this area, and it's a really important, great paper. He was also hilarious. I think there's videos online. If you go to YouTube, he's hilarious to hear him talk to. He told really good stories, the character. So anyway, so that's an example, but your eye does stuff like that too. We don't have bug detectors. Bugs aren't so important for us, but there are lots of different systems in your eye that seem to be taking information, processing, and then passing it down through the optic nerve. So one anchoring point I can give you is the ganglion cells, again, the last layer before you go into your optic nerve. There's 21 different types, last I heard, that do something functionally different. We don't know exactly what they all do, but they functionally they have different physical architectures, and they functionally seem to be transmitting different kinds of information. The other thing to remember about your eye, and any eye, is it's event-based. So when you train a network or any computer vision system, you typically give it either a single frame or you give it a series of frames, but it's all very discrete. Your eye doesn't work that way. It's completely asynchronous. All of your receptors are firing. They're sending information down through those layers in your eye, and they're passing them on to the optic nerve in an asynchronous manner. So the information flowing through different points in your optic nerve through those million samples can all be at different time points in terms of when they hit the retina and when things fired and came through. And so that's continually coming through at this very high frame rate, and you have to process it. But again, there's not a discrete frame that you can process in any particular way. So people are interested in event-based computer vision, but it's hard for all sorts of reasons you can imagine. And how you would take a deep network and turn it into something that can process event-based information is also really hard. Questions? Okay, so then you get into the brain. The first way station in the brain is called the LGN, or lateral geniculate nucleus. You don't really need to know that. You go from LGN, which does a lot of processing, to V1, which is your first cortical vision area. It's back here, okay? And there's this clever set of naming conventions where you go from V1 to V2 to V3 to V4, okay? But the weird thing that happens is you go from there, you get a 400 to 1 data expansion. So you go from 100 million samples on your eye to 1 million samples in your optic nerve, up to by the time you get to V1, about 400 million samples, or 400 million discrete neurons interacting. It's kind of crazy, right? There's this weird compression and then expansion. And there is actually, in the deep learning literature recently, a couple nice papers looking at this kind of compression, specifically in the retina into the brain, that suggests that that bottleneck is really critical, and that bottlenecks are actually quite advantageous for learning. And that in deep network architectures, bottlenecks could also be very important. So now I can point people at those particular papers if they're curious. So then you move along what's called the ventral cortical stream through your brain. The number of samples increases once again. You get about 10 to the 9th neurons in so-called higher level visual areas. It's sometimes called inferior temporal cortex, IT. Okay. When we look at how those neurons respond to information, which you do by sticking an electrode in and showing the animal pictures and then figuring out what the neuron fires to, very straightforward, you get a feature hierarchy, okay, a lot like a convolution neural network, that you've got a feature hierarchy. You go from simpler features to more complex features if you look at the visualization of network units. But there's also a lot of feedback, something people have just begun to become interested in more within, deep RL, recurrent networks, and so on. But we have about twice as many feedback connections coming from the top of our brain back into your visual system as you have going forward. And we don't know exactly what those feedback networks do. Something about attention, something about prediction. It's not at all clear. It's very hard to ascertain, but there's a lot of feedback coming back through the system. Okay, and just to give you a sense, your entire brain is about 10 to the 11th neurons with 10 to the 15th synapses. So that's the parameter space you're dealing with. Maybe it's larger than that, though, because there's all these chemical systems as well. And each neuron is connected. They're not fully connected. They tend to be connected to about, at most, 10,000 other neurons. But the kinds of connections may be functionally different from one another. So there may be all sorts of interesting information being coded in the way the connections form as well, not just the fact that it's connected. So there's a large amount of complexity there. It's a big space. Questions? Interesting?
Has there been any work to look at, because I know that when we try to look at these neural networks, usually we use like simple activation function. Has there been any work accurately model all the complex phenomena that goes on between the synapses with the different neurotransmitters, or like you said, the different types of connections in different areas?
Yeah, so actually Carnegie Mellon has one of the best computational neuroscience programs in the world. And there are a group of people here and at Pitt that specifically look at that at the circuit level, trying to understand how circuits and the actual neurotransmitters and things end up creating the complexity of information processing we'd expect. That work is really hard to connect to the higher level network architecture stuff we often talk about. And it tends to be in very simple circuits, like a very simple motor cortex circuit or something. But I think there's always been a group of people and a continued interest in trying to understand that. And some people argue we're going to ultimately need to build neural networks that incorporate that kind of information processing as well, that other layer. There's also people which I would characterize a little bit on the fringe that believe that you have to actually get down to the physics level, that quantum is actually important too. It seems a little far-fetched to me, but we don't entirely know how information processing works, and it could be that there's stuff happening beyond chemical that we don't completely get. So it's a good question. Okay, so here's the problem if we think about it at a higher level, though. I love this cartoon. I think for most science it applies, actually. So if you can't quite read it, it says in the middle, then a miracle occurs between the two sets of equations. And he's, should be a little more explicit here in step 2. So we're pretty good at characterizing inputs. And I can tell you a decent amount of how the retina works. Not everything, but basic response to light, how it's coding information. I can tell you a lot about how the information gets organized in the early visual areas. And I can tell you a lot about what's happening in output. I can tell you about behavior. I can tell you about the kinds of categories we learn about. I can tell you about the way those categories seem to be coded at the higher level in the brain, like where information about faces seems to be processed, where information about scenes seems to be processed, about hands, about tools, about living things, non-living things. But that's all like, okay, we get input, I can characterize input a little, and I've got output, I can characterize output a little. All the stuff in the middle, all the feature hierarchies are a black box. Now, that's not unlike, of course, deep networks. We can characterize the inputs pretty well. If you look at the early layers, it's fairly clear the kinds of features they're coding for. As we'll see in some of the visualizations I'll show, but in general, it's really hard to characterize what's happening in intermediate layers. Then you get usually to your last fully connected layer, and you look at the units. It's pretty obvious what they're coding for, and then, of course, at output, it's completely understandable. So there's this interesting problem that recurs a lot in science, that inputs are easy, outputs are fairly easy, and all the stuff connecting, it's hard. And that's where we're kind of at in neuroscience right now. So what do we do to try and address that? Well, there's lots of different ways of collecting brain data to try and figure out what's going on in the middle. You can do a brain parts list. People try to do that. You just want to characterize all the different kinds of neurons in the brain and how they're all connected and chemically interact with each other. That'd sort of be like if you're trying to explain how deep networks worked and you did a parts list of all of the different parts of the CPU and the GPUs and all the power supply and the cooler and everything else in your server. Wouldn't get you very far, but some people worry about that. There was a big project a couple of years ago that got a press called the Human Connectome Project. I don't know if any of you have heard of it. The Human Connectome Project was an attempt to define the connection matrix of the entire brain in terms of a particular set of kinds of connections called white matter fiber tracks. These are sort of longer range connections, not neuron to neuron connections, because all neurons are assumed to be locally connected to lots of their neighbors. But there's these longer range connections which seem to be like the wiring diagram or the breadboard connections that allow information to flow in interesting ways besides local connectivity. And so you can actually download all of the human connectome data. I think it's 1,000 subjects. So you can get a full wiring diagram for the human brain across 1,000 people and play with that data. It's sort of helpful in some cases, but not completely informative about how the brain works. It may be useful, we'll see. You can do brain activity maps. So you can record the activity of all of the neurons in the brain at once, or specific neurons. So this is sometimes called functional information, because it's about doing a task and then how neurons respond to the task. So as I mentioned, you can stick an electrode in. You can record from a given neuron when there's some stimulation, something driving it. You can also do MRI. Most of you have probably heard about doing MRI. In that case, you record from every neuron in the brain, but at a much more coarse level. I'll show you in a minute what I mean by that. And you can see the response of all those neurons across the whole brain as people do a given task. You can use EEG, which uses scalp electrodes, and you can record the summed electrical activity at the scalp of all of the neurons in your brain in a moment by moment in time. You can use MEG, which is another scalp electrode sort of system that also records basically electrical activity at the scalp. Notice there's no way to get in there and record from all of the neurons simultaneously. If you record from specific neurons, it's only a couple. The best you can do is maybe 1,000 with these weird little things they call a bed of nails that you push into the brain. You're not going to, but again, we have hundreds of millions of neurons. And then if you record from hundreds of millions of neurons, you're using things that are outside of the brain. It's sort of like cosmology. We don't get to look directly. We have to sort of have our version of the Hubble Space Telescope looking at things. You can do behavioral prediction analysis. I think sometimes people forget that's a very useful tool for understanding how people think. So you can build predictive models of the way people behave. Don't worry about the neurons at all, but you can understand at the cognitive level what's going on. I think people in HCI do that a lot, like the password security people do that. I think some of the LTI people do that. And then you could also make connections to other data sources. There's genomics, the computational biology people do that, proteomics, behavioral economics. If you want to go the other direction to macro level behavior, like you might want to predict, for instance, the behavior of the entire world's economy based on what's happening with the coronavirus or something like that, which would be very macro scale, but it might be useful to have AI systems that can predict at that level and look at human behavior. So sometimes we call this computational social science now. Like Google had this thing that turned out to be kind of a little bit of a disaster, but the flu predictor, if some of you have heard about that, where they were looking at the kinds of queries people were making on Google as a way of predicting where the flu was occurring. But you could use data like that as well to understand things. Okay, I'm going to focus more though on neuroimaging. How are we doing? We go till 10:20, is that right? Cool. Okay, so there's some challenges with neuroimaging. We'd like to be able to look at the brain. One is it's often expensive. Okay, to anchor an MRI, even if you run it with your own MRI machine that you own on campus, like CMU does, is somewhere on the $500 to $600 an hour range to run it. Okay, between the cost of the scanner, the maintenance fee, the electricity, which it uses a lot of, the technician to run it, those kinds of things. There's a lack of power, and by I mean here statistical power. So the number of observations you can get in a given study. Let's say you're doing a human and you're doing an MRI study or an EEG study or something, you're probably only willing to sit still and do the task for something like an hour to an hour and a half. Much beyond that, you're going to go completely crazy. So there's a limit in how many observations you can get. And also the number of individuals, like the scale at which you can collect data from people. So one thing you can do is you can try, if it's behavior, you can crowdsource, use Mechanical Turk, or that's what the CAPTCHA system is doing now. They have people Google has people labeling things as a way of collecting data from all of you. But there's a limit often in the total number of data points you can collect across individuals. You've got this problem that people and brains are different from one another. So you have to have some way of aligning all of your data. So people don't think about that. We all look different. We have a lot of other personality differences. Our brains look different from one another also. So if you want to talk about something aggregate across the way humans are, and you're looking at brain data, you have to have some good way of aligning all of those brains. And it's harder than you think to align them all. There's some landmarks and some obvious things to do, but it's not perfect. And if you're talking about precise, different parts of the brain about how they process information, alignment might be critical. So that's a big challenge. It's actually some of the people in machine learning are very interested in this problem. How do you take data from multiple sources and align them all into a single space? You've got this high-dimensional data set. You've got lots of neurons. You've got lots of data points. We don't really know the dimensionality of the data set in terms of the underlying critical dimensions. Right? If you're talking about visual space and learning about inferring about the visual world, what's the actual dimensionality of that? Is it categories? Is it other kinds of visual information? We don't really know. So you've got this high-dimensional data set. You need to reduce it down to some lower dimension, but the structure is completely unknown. So that's a big problem. And then as I'll show you in a second, there's also a big trade-off between spatial and temporal resolution and invasiveness, meaning how much you poke into the brain versus do it outside of the brain for studying things. So here's a nice diagram I found on the web. Trade-offs in neuroimaging. So you can see, let's see, I'll pull up my little pointer. I referred to EEG. EEG is a system where you have these scalp electrodes. It's relatively inexpensive. They don't have cost here. It's also not invasive in the sense that you're not sticking anything to the brain. The worst is you get some gel in your hair. If anyone wants to be an EEG experiment, we run them in the psych department. You put this weird scalp cap on, and it has these little electrodes that get gelled to your scalp, and then you can record brain activity at the scalp level. Notice its temporal resolution can be pretty good. You can get down into a fairly fine-grained level. Scalp ERPs, which is a variation EEG, you can get down almost into the 1 millisecond level in terms of resolution. So the signal's changing at that rate, you can pick up the differences. But spatial resolution, in terms of what you can resolve within the brain, is really crappy. They've got it over here between 10 centimeters and a meter. I mean, that's like most of the brain. So you can tell like it's happening on the left half of the brain or the right half of the brain, but not much more than that. That's actually a little bit of an exaggeration. You can get down probably into the couple, like 3 centimeter region with these systems. Meg is a little better, maybe. It's a lot more expensive. Meg may also just be hallucinating better data. It's not clear. They make pretty pictures of where things are happening in the brain, but I'm not sure if it's just a hallucination at this point. Now, one thing which we talk about a lot is fMRI. Again, MRI is expensive. It's not really invasive in the sense that you lay in a scanner, you've got this like Iron Man helmet on, so it's got a head coil so you can record from your brain accurately. It doesn't hurt you, it's just a big magnet. There's no evidence that big magnets hurt anyone unless you happen to have metal somewhere in you, and then that's probably bad to be in the big magnet. But it's got pretty good spatial resolution. We can get down into the 1 millimeter range now. You can resolve things at a level of 1 millimeter. To give you a sense, a cubic millimeter of brain tissue is about 10,000 neurons. So you can't resolve single neuron activity, we can resolve activity at the level of 10,000 neurons. On the other hand, fMRI is really, really slow. You're talking a second to minutes even. Anyone know why it's slow? Any guesses? No one even wants to guess?
Because you have to scan and then analyze the whole thing?
No, it's not a pipeline problem. The scanner, well, there is some rate at which the scanner has to go through the whole cycle, but that's relatively fast. It's called the TR. You could actually push your TRs on these big magnets now today at the level of like a second. So you can go pretty quickly. If you want to do a smaller part of the brain, you could get down to less than a second. So it's not actually a physical device. Any other guesses? Anyone know what fMRI is measuring?
Isn't it like magnetic response to
Magnetic response to what?
oxygen
Oxygen. Where is oxygen?
in your blood?
In your blood. Okay. So there's something in MRI, it's measuring something called BOLD. BOLD oxygen level, or blood oxygen level dependent response. Okay. You're not measuring neuroactivity in fMRI. All those pretty pictures of the brain you see with the colors all lit up, they're showing you actually something about blood flow in the brain. Now, it turns out that blood flow and neuroactivity correspond quite nicely in the sense that when you think and you use up metabolic resources, your neurons firing, you need to get more blood in there quickly to replenish those resources. So there's a lot of evidence now that it's a reasonable proximal measure of neuroactivity, but there's a problem. Blood is slow. The neuroactivity happens in the order of milliseconds, but the blood flows in only on the order of seconds. And in fact, the whole hemodynamic response, the response function from when the blood is called for in that part of the brain, because you've used up metabolic resources, to the point where it goes back down to baseline, is something about like 14 to 16 seconds. Now, the way the curve looks is really, the peak is about 6 seconds, and by 10 seconds, you're getting pretty flat, but it's still not zero. Okay? So your ability to resolve events is only as good as the fact that your responses don't overlap with one another, right? If you have two responses overlap, you'll never resolve them. So if you're measuring something where the response times are in the order of 10 seconds, you have to have your events on the order of 10 seconds. If you make them shorter, you have to do tricks to try and resolve things, but you're making guesses between overlapping functions. It's pure statistics. Everyone understand that? So fMRI, our favorite tool for human neuroscience, is really slow. It's resolving things in a really quite slow way. That means that the number of observations you can get for your $500 an hour of a person in a scanner is usually on the order of 200. So I spend $500, I get you to look at 200 things total over that hour. That's not very big when you think about like the way we train data sets, the way we use data sets to train these days, or what we'd like to think of as large-scale data. Okay. So it's a real challenge in terms of what we've got. And where we'd like to be, of course, so we'd want to be over here, high spatial resolution, down, forget about 10,000 neurons, we'd be down like at the level of a couple neurons. And we'd like it to be very fast because neural events happen and your mental events happen in the order of milliseconds. We know, for instance, that you can detect a spot of light and respond in about 50 milliseconds. We know that in 100 milliseconds, your brain is capable of identifying the scene. Maybe not in fine-grained detail, but you can tell what the basic scene is, if it's outdoor or indoor, if there's an animal in it or not, all within 100 milliseconds using other techniques. So we'd like to be here. We don't have a technique really for going in here. You can see single unit recording. That's where you stick electrodes into the brain. You can do that with animals. You get very, very few neurons, but you get good spatial resolution, good temporal resolution. But it's highly invasive. So you're not going to do it with people routinely. It does happen with people. There's times when people have epileptic seizures, so they're going to be doing neurosurgery. And so to localize where those seizures are, they're putting electrodes in anyway. So there are groups that then have done experiments while you've got the electrodes in there. But as a rule, you're not going to take your average undergraduate in a class and say, by the way, would you like to volunteer to have some electrodes stuck in your brain? Okay. Parents frown on that when they come sending kids to college. So there is a real challenge in what we can collect about brain data. And I think it's one of the major engineering challenges we have. We have a lot of ideas. We'd like to connect it back to these large-scale models, and we're just data stuck. Actually, I have this idea. We collected a big data set in the lab this past 2 years now. We had 4 subjects come to the scanner 22 times each for an hour and a half each time. And we ended up collecting 5,000 data points for each of them looking at pictures. They actually were looking at pictures from ImageNet and COCO, from computer vision data sets. That was pretty heroic. It's a lot of work. It's 100 hours of scanning basically the time we were done, because we also collected some anatomical and some other information about their brains. Someone else in Minnesota, Kendrick Kay, recently did 8 subjects for 40 hours each. I think some of his subjects are like never going to get within like a mile of an MRI scanner again. But those were heroic efforts to try and get a couple subjects in, plus it was really expensive. So what I'd like to be able to do is crowdsource neural data. Ideally, you'd be able to get people to all run small scans for like 15 minutes while they're doing some other study, and have some way of it aggregating all together and us aligning it in ways that we can learn things by having some overlap between all of our subjects. And so it's a project we're going to begin to do some piloting on this summer, and I'm hoping ultimately we get to the point where instead of one lab heroically trying to collect a lot of data, you have lots and lots of labs collecting a little bit of data, but you put it together and you get a lot of data. But
Are there any advancements happening in the neuroimaging technology itself?
yeah, well, that's an interesting question. People have tried to improve EEG. There's a group here in engineering that's been working on that. We've been working with them. People keep hoping that we'd be able to measure electrical activity directly using a scanner, an MRI scanner of some sort. That hasn't really happened yet. People keep pushing. There was also this guy at Berkeley that had this idea of neural dust, where you inhale these little nanobots, and then they latch onto all of your neurons, and they have little radio transmitters on them, so you could record from all of your neurons simultaneously. But that was kind of science fiction. He never really, he wrote a proposal to try and get it to work, but they never really got it to work very well. So right now we're kind of stuck. There isn't an obvious breakthrough on the horizon. Things get marginally better, but not obviously. I mean, ideally we'd want to measure something other than blood in the brain, but non-invasively. Again, using a big magnet or something else that doesn't hurt people or animals. So that's a good engineering problem. The people that invented MRI won the Nobel Prize a couple years ago. Okay, so good. All right, a little bit of background here, and then I'm going to talk to you about some neuroscience. There's this long-standing underlying assumption that vision is compositional. What do I mean by that? It means that your notion of objects, scenes, whatever things you think about are built up of these separable parts or building blocks. It's a bit like Lego. The idea that you can decompose things into these parts, and the way your visual system works is that you're learning about and learning to extract these parts and are ultimately assembling them to describe what objects are. And I mean, the easy example is always, well, a face is composed of eyes, nose, mouth, and so on, and that you can recombine them in different ways to get new faces. And like, for instance, wheels can appear in lots of different things, and you know about wheels separately. Okay, so this recombination is really critical, and that these parts are specifically a consequence of that progressive hierarchy. If you look at the earliest levels of the hierarchy in a convolutional neural network or in the visual system, you start with these low-level features, edges, contours, but you move up to the point where you have higher-level representations that cover what look like parts of objects but not the whole objects, and those are somehow recombined ultimately to describe objects. So that's like this long-standing belief about the way visual system works. And again, that's reflected in modern neural net architectures. So what has visual neuroscience done? They've often focused on the nature of such features. What we really want to do is explain how you get the early level features, how those get hierarchically built up and recombined into bigger and bigger and more complex features, and those become objects. That's been like the name of the game for a long time. Okay. There's this really great story about that kind of thing. There was Charlie Gross was an early neuroscientist at Princeton, and they were doing recordings from the inferior temporal cortex of monkeys, and they were trying to figure out what the neurons did. And this is like early in the days of neuroscience, and they really didn't know. And they were showing all these different things to the monkey, different kinds of patterns. They had found that some neurons were selected for faces, but there was this one neuron that they were stuck on. They kept showing it different things, and they couldn't get it to fire, no matter what they did. And then someone randomly pulled out a toilet brush and showed it to the monkey, and the neuron went crazy. Okay? And so it's a true story. They took them a while. They looked at it. It was this toilet brush neuron. You're like, why would there be a toilet brush neuron? The monkey had never seen toilet brushes. It must be some kind of feature of something else. Later on reflection, what they realized is all of the people, this is in the 60s, and Charlie was a bit of a hippie, had fairly big bushy beards. And it turned out that the monkey, of course, had been reared and taken care of and been fed by people with big bushy beards all the time, and was probably somehow coding for some of the features in the beard-like pattern. So the faces they were using as stimuli weren't their own faces, they were faces that were beardless. It didn't like that very much. This neuron seemed to like the beard-like pattern. That's a guess, so they have no way of really knowing. Another thing that they do, which I'll talk about, is there's image reduction. So you can start with a complex stimuli that the neuron seems to respond to, let's say a full picture. And then you can start to reduce or remove things from that picture and see if you can sustain the response of the neuron still, right? So ideally, you should be able to remove stuff and continue to get the neuron to fire, and then eventually you get to the minimal stimulus that still drives the neuron. And that's sort of the idea that that would be the feature that it's really selective for. So there's been some work on that. That's hard to do for all sorts of reasons we'll talk about. And the last thing people do is genetic algorithms, where people try and evolve stimuli that drive the neuron optimally by moving down some kind of genetic hierarchy. And I'll talk about that as well. Okay, so here's an example of image reduction, probably the most famous one. This is by Keiji Tanaka. They started with neurons that seemed to be selective for all the stuff on the left of the arrows. Sometimes weird shapes, but still, recognizable in some form. And then what they got in the end was these patterns on the right of the arrows that were the things that the neurons seemed to be still sustained responses to. Now, here's the problem with what they did. They did this completely ad hoc. It wasn't evolutionary in any sense. What they did was they basically tried removing parts of the stimuli or the complexity, and if it didn't work, they tried removing something else and sustaining the neuron response. And they kept doing that through the whole process until they got to whatever was on the right. So they could be doing some kind of movement into a local minima. It could be that there's a lot of other trees that they could have gone down that would have also sustained the neuron's response, so they don't really know. Right? So it could be, for instance, here, this one here, well, they come to this, but maybe they chose to pick these two regions and really move down that pathway, so they got this. But maybe if they turned out they'd only kept the feet in some part here, it would have performed equally well. So this could be a completely wrong mapping of what the minimal stimulus is that neuron drives. There was no real principled way of doing it. It was mostly intuition. And actually, a lot of science is intuition, I have to tell you. more than people realize, I think. Not just neuroscience, but chemistry and other things. You have a kind of implicit hypothesis about what you think is happening. You change your conditions to see if things move the way you thought they might move, and then you keep trying that. But it's all based on your own intuitions.
Do we know if a minimal stimulus is guaranteed to exist at all?
No, and that's an actually really interesting point I'm going to get into. It could be that the actual stimulus really is just the whole thing, or something that's not minimal really, but it's more complex in some way. So that's the other problem, is your starting point, what you chose to show the animal, that said this neuron's, and I'll show you some data in 1 second about that. It could be that there's something else out there that would drive the neuron much better than anything you tried. And you have no way of knowing that. So one big interest, by the way, though, at Carnegie Mellon recently is what we call automated science. And the idea that for a lot of these sciences is there's some way we can use modern machine learning and search as a way of optimizing search through a high-dimensional parameter space rather than just sort of randomly going by our intuitions. So that could be true in chemistry, biology, lots of other domains too. So this is actually a good example of what you just asked. So this is some data from my friend David Sheinberg's lab. So it's a little hard to see the pictures, but each row here is a neuron. So this is one neuron, another neuron. Okay, the stimulus on the left is the top-ranked stimulus. So this is a stimulus picture that drove that neuron the best of any picture in the thousands of pictures he used. Okay, and that's the response, that black line. Okay, the trick is the ones on the left are always things that were familiar that the animal had seen a lot of and had been trained on. The ones on the right are ones that the animal had never seen before, but were shown for the first time, and then this is the top ranked one for things that were unfamiliar that drove that neuron. So you have two sets of rankings, for familiar on the left and then unfamiliar on the right. Okay, so you can see here for some neurons, there's a couple things to notice. One is that the top ranked stuff doesn't look very similar to one another. So it would be really deceiving to do some kind of minimalization technique to find the optimal stimulus with any of these things, because there's clearly a lot of selectivity in this neuron that's not being explicated by what's going on. Question?
How do we know which one, we should record, like which neuron to record?
You stick your electrodes into a place roughly, so these were recorded from inferior temporal cortex, a place we know is object selective. That's all we know about it. You know, you just pick a neuron. You got a neuron. There's no way to know. We don't understand. First of all, we don't know the functional basis in the first place, and the accuracy which you can put these things in is low also. So you just have some large, you can pick visual system or auditory system or motor system, but other than that, it's a crapshoot.
There's a lot of way to manipulate the result. That's what I'm hearing. I could have gotten some other kind of graphs from other part of the same region. Maybe.
Right, which is why he's got lots of neurons. So his actual, I think in this paper they recorded from thousands of neurons over a long period of time. And one of the things you see is that for this level of the brain, there isn't a lot of consistency. I mean, this is ice cream cones, but other random stuff. This is, I don't even know what those things are, bananas and other things. Right, and then if you look at all 1,000 of them, they all have that kind of characteristic. So they were all kind of idiosyncratic relative to each other. So part of the point of this, you don't learn a lot from this kind of endeavor. So yeah, it's not that you can manipulate the results so much by what you pick, because we're not that precise. What you could do is you could pick and choose which ones to show. But usually the idea is in a paper you show all of them. But I think it's true both for this kind of work and for like the neural network. People tend to visualize the units that are most interesting. So when they publish deep network papers too, they always show these particular nice examples. Oh, this is like an eye unit or whatever, but they haven't shown you the many, many other units in the network that look really weird. Who was it by? There was a really good paper that also had an associated website recently, the Google group, where you can explore yourself the activation of every unit in the whole network and look at the gradient of coding. I can't recall the name of the person, but it was a pretty cool way of looking at that to see actual performance a little more carefully. Because this is a later slide, but I'll mention now since you raised it. There's a problem that people like to visualize units within neural networks. And then they make a lot of inferences. Oh, this is an eye unit, or this is a face unit, or this is a dog unit, or whatever it is. And I think that's a really, really terrible idea for exactly the reason you named, that you're being very selective in which units you visualize. And also probably visualization is a terrible metric for deciding what the units are doing anyway. because they're actually just units that are solving a task in the context of a large number of units as intermediate stages, and there's no reason why visualization or us understanding them spatially should be a metric for whether or not they do the task well. So inferring parts is a really terrible idea, which is true, I think, for the neuroscience. I think it's very symmetric. So that leads kind of nicely. So trying to understand the system, where a miracle occurs, there's been somewhat frustrating progress. We can tell a lot about early visual system because there's a lot more consistency. If you go into earlier visual system, V1, V2, what you get is you get a set of oriented edges. And it's very systematic. So there's a set of edges, or basically contrast boundaries, and they tend to be, okay, you've got an edge that's like this direction, then this direction, that direction, and so on. It's very systematic across all of V1. Neural networks look a lot like that too. There isn't this kind of idiosyncratic problem when you record from different places, you get different results. It's very clear. But when you get up in these higher level layers, you get very fuzzy results. So at the area of IT, you get these areas that are selective for categories. But there's a term I want to use here. How many of you have ever heard of the face area or the face network in neuroscience? It's in the popular press a lot. No one, maybe? Okay. You'll read that a lot. If you read the New York Times or other things, whenever they talk about how the brain works, they'll often talk about an area of the brain doing this or whatever. That's what I call reification. When they say this is the face area of the brain, what they're saying is this part of the brain responds more to faces than to other stuff. It's selective for faces somehow. But that's not an explanation for what it's doing computationally, right? That's describing the fact that ultimately its computations are sufficient to differentiate faces from other stuff. But that doesn't provide an explanation for how it ends up doing that. And so that's this term reification. The empirical definition of it can't be the empirical phenomenon itself. So it would be very unsatisfying if what you did is you said, oh, I have a deep network, and here's the face parts of the network, and that's what it does. That doesn't describe anything about what the network's doing. You couldn't build a network based on that. So we have to be really careful about thinking that when we describe the selectivity of something, that in itself is an explanation for the selectivity. Again, artificial systems and biological systems. There's other problems there too, but we don't have to get into the details. Okay, the other thing is the kinds of features that we tend to find, like I showed you in that previous slide from Sheinberg's lab, don't support compositionality or the kind of generative process that we kind of expected or hoped to have. It's not like a CAD program where you can take all the 3D parts and put them together like Blender or something into something more complicated. There seem to be kind of parts, but they don't combine in ways that are intuitive or obvious to us. So one of the things I think comes out of this is maybe, this is about the visualization point too, maybe inspection about the way things respond is the wrong metric. Our sort of conscious inspection or intuitions is maybe not a good evaluative metric for whether or not the system's doing what we need it to do. And that's true again in neuroscience about looking at how a neuron responds or us looking at a unit within a network. Something to keep in mind. I'm not sure we have the answer. So we need to collect a lot more data. We need better search algorithms. We need to run better simulations. One of the things that's happened, and I'm going to try and go through this fairly quickly, is because deep networks have gotten to the point where they're very high performing, there's been a lot of interest in pushing them back into understanding biological systems, and there's been some promising results that begin to help with a lot of these problems. I already mentioned this. This is a visualization problem. I sometimes call it stupid CNN tricks, because people get all excited about visualizing things and then claiming that they have particular functions. And so this was drawn from a Google paper where they made all sorts of crazy inferences about these pictures on the right and called them objects. So the ones on the far right were objects, the ones next to that were called parts, and they claimed that all these units that respond in this way, that's what they were. And it's a reach by far. But what I want to talk about for the last part of the lecture is some recent work that has tried to push the idea that convolutional neural networks can be good models for biological vision systems, and that we can understand something about both in both directions by doing that. So again, there's this idea that convolutional neural networks were inspired by the architecture of the visual system. And you can see on the top row there, there's a map way of the brain going from the back of the brain, V1, forward into these inferior temporal areas, PIT, CIT, and AIT. And there's a kind of hierarchy of layers that you might expect And then there's a very simple convolution neural network down there, which has a similar kind of architecture to it. So it's something people took seriously, people tried to play with. So what Yeamans and DiCarlo did was they said, goal-driven networks are good neural models. And they said, well, regardless of the parameters they have, oh, sorry, do you have a question?
Oh yeah, I had a question actually about whether there is a lot of noise in the data that you collect. When you collect this biological data, is there a lot of noise?
Yeah. So MRI in particular is very noisy because blood is noisy. One of the big things you're always focusing on is a lot of signal processing problems to try and increase your signal to noise ratio in various ways. Physiology direct electrode recordings aren't as noisy. But even there's a bit of hallucination to make it clean itself up in ways that are probably not always warranted. So that's one of the things you do is you need more data. So we're challenged in our ability to collect data, but the way you'd overcome the noise is by collecting more data. So we always have that inherent problem.
And then kind of a related question, when you measure the, I guess it's like the electrical impulse on the neuron, does it actually affect like, the current on the neuron so like it might not actually fire out?
Yeah, it's one of those physics observer effect type things. It kind of does. But one thing, you're ultimately killing the neuron by putting an electrode in. So eventually the animal won't have that neuron anymore. So I better not be the only neuron doing that. But the theory is you're not actually affecting its responses while you're recording from it. But that you are killing it eventually. You lose the neuron. Lose the neuron is the nice way they put it. So the animals seem pretty much fine. So I think that there is a lot of redundancy. There's no such thing as a super sparse code where a couple neurons are probably coding for whatever it is. So just quickly though, the point that they're making is that maybe if you have a system that solves the same task that a biological system is solving, you end up algorithmically coming up with the same solutions. So high-performing models that solve a task well would be good candidate models for also explaining how the brain works. That's an important point. As you optimize performance, you're also optimizing explanatory power. That's the argument they make. Now, that could be wrong, right? But so here's what they did. They optimized network parameters for performance. So they took some ecologically valid task. In this case, it was object classification. They tried to make a high-performing network to begin with. And then what they did was that once they pushed it there, they fixed the network parameters, and they compared it to a network to the neural data and tried to see how well it could explain the data. Now, what you're going to see is they went through that space multiple times in terms of optimizing and then tried to predict the neural data over and over again. We can see the trajectory of what happens when you change optimization. And it's a lot easier than neural fitting. So if you did neural fitting, you need a lot more data. So it's a lot easier to just take a lot of human labeled data for the task, right? Solve the task problem, optimize a network for solving the task, then apply to neural data, then getting enough neural data that you can fit it network directly to the neural data. Because we rarely ever have enough neural data to take a network and fit it directly without some kind of constraint on its performance. Does that make sense to people? Okay. So, okay, we can skip that. So the argument is that neural-like models do optimize performance. So here we can see, again, the architecture of the visual system, the recording from it, the recording in these sites right in here. Okay, this is the network architecture they use. It's not really that important. It's actually an older architecture at this point, the way rate things move. But the key thing to see here is that this is, these are performance on the task. This is the neurons for performing the task if you use them as a predictive values for performing a given task. This, again, object classification. This is neurons from a different region of the brain higher up. This is humans performing the task just behaviorally. And then this is their optimized network, what they call HMO. And they were comparing it to a bunch of computer vision algorithms, some of which you may know about SIFT, HMAX. But this is the convolution neural network here. And you can see this high-performing convolution neural network actually performs the task as well and comparable to humans in each case. So it's optimized well. But this doesn't tell you whether or not explaining neural data. This graph can show you that. So what we have here is we have 3 populations of models. Each one of these points represents a model. Okay, and this is how well it performs. So as you move further to the right, the model's performing better. The blue ones were ones where you tried to optimize performance of the model first, and then you fix the parameters, and then tried to predict neural data. Okay, the green ones where you randomly selected things, weights, and then you just let it run. And then the red one was where you tried to fix the model parameters only after you tried to explain as much neural data as possible. And then you looked at how it performed. Up here on the y-axis, you have how much of the neural data was explained for individual neurons recorded for the monkey. And again, it's not a large population of neurons. It was a couple hundred, I think. And what you can see is that your best results come when you first optimize the performance of the model, and then you look to explain how much of the variance in the neural data you can account for. Now, there are a couple small cases where you get slightly better neural explanatory power, but your model performance is much worse. So the interesting thing is there is this progression, as you might predict. As model performance improves, you actually improve your ability to explain neural data. Now, here's the cool thing. You then take your best model from here, your highest performing model that also optimized as much as possible the neural data. And then what you do is you start there, I think it was this point here. And then what you do is they have another way of optimizing over both parameters, and they keep moving in space, in that space. And what you can find out is that once you have that best model from that overall thing, where you allow model parameters now to vary, you can improve model performance a lot more, and you also improve your ability to explain the neural data. So higher performing models are really the best at explaining neural data. These are also old computer vision models. So this is about the best that they can possibly do. They don't really have a lot of model parameters that can learn. And they can only get so far in explaining neural data. Their performance is fairly crappy. You can get both good performance and good neural data explanatory power. So it's a pretty cool paper. It was an important paper. Just show you, they can also explain individual neurons. So this is a complicated graph, but basically what I can show you is for each of these is a row. There's individual neurons being explained by taking the pool of features from within a given layer within the model. You can actually explain individual neuron performance, which is great. And I'll show you the same thing works with human fMRI. This is a different group. They're looking at the data in a slightly different way, but what you basically do is you can create a similarity matrix between all the stimuli shown to a person, and you can look at the similarity matrix for each pairwise combination of all the stimuli. This is grouped roughly into artificial objects, natural objects, faces, bodies that were not human, and faces and bodies that are human. And you can see the similarity matrix. So obviously, you always have the same image. It's always going to be highly similar to itself. But you can see strong similarity in responses in MRI when you show a face to a person, even when it's a different face. You can see similarity matrices here. Again, when you look at, for instance, natural objects and you compare them to other natural objects, you get high similarity and so on. What you can see is here is the network response for a model, a hierarchical neural network model that was optimized again for performance, and it creates almost the same similarity space in terms of how it represents objects. It depends on which layer you're using. You get better and better responses in terms of explaining the similarity between these two graphs as you go up into the higher level layers of the network. There's various other details that we don't have to get into. You can look at the paper if you want. I've got like 5 minutes left. I want to just show you, this is a summary. I want to show you a couple things quickly. One is sometimes people have sort of complained about neural networks because they're bad at semantic interpretation. This is a fairly famous paper that came out. Some of you may have seen it. These are the captions that were given for describing these particular scenes. And they're obviously horribly wrong in each case. But I don't think that's particularly fair because almost all the neural networks were designed to solve a very particular task. In this case, a particular captioning task it was tried to perform was done on all normal pictures. Suddenly you give it these really weird pictures that have different context, and it fails horribly and gives you these bad descriptors. Okay? But it's not actually unreasonable. It knows about tarmacs, it knows about airplanes, it sees the two, it goes, oh, it's a plane parked on a tarmac. Why not? Okay? We have a lot more context and information than the networks do. And this again gets at a problem that the networks only do as well as the labels. We know a lot, and one of the general AI problems that we don't know how to solve yet is the fact that we can bring all that context to bear. So when we look at this picture, we go, that plane's in a really funny position and shouldn't be that close down to a road. That's really horrible, and it's also at a funny angle. Therefore, there's something really bad happening. Okay? Or here, that house shouldn't be like that right next to the beach then that way. But there's no way that the network could figure that out without a lot more contextual knowledge. So to complain about a network not doing well or saying it's not human-like is kind of a cheap shot. It's like low-hanging fruit because of the fact that we bring a lot more to bear that the networks don't. Yes?
I was actually going to ask you about that after, but I was wondering about that as it relates to labels as well, because like you were giving the example of that you have a kid kicking a soccer ball outside, and all that, all the network is seeing is what the final label is,
Right.
And so it doesn't have the context to bring in the outside and all
Yep.
the other models that we have.
Yeah.
Has there been any effort tried to attack that with like hierarchies of models or something?
Yeah, people are trying to do a lot more of that. But bringing in context is still one of the harder problems. But I think also we need to move to unsupervised methods. I mean, we should be learning a lot of that context. There's probably something that's occurring over and over again in particular kinds of like ball-driven sports that you can learn and infer things about, even if you don't have explicit labels for them. And that kind of underlying structure, I think, is where a lot of it's going. So at NeurIPS this year, a big theme across a lot of different groups, including some of the major groups, was unsupervised learning that's biologically inspired. Trying to get better metrics that look like kind of predictive, contextual things people do, understanding more about the biology to do that, and doing it in a more unsupervised way. I think that's something where people realize that the whole field is going to have to go, partially because the labeling approach is kind of reaching the limit where we can eke a little bit of a better performance by putting more layers in ResNet 10,053 or whatever you want to call it. But it's just eking a little bit out. We're getting to the point where the next steps are going to require new architectures and new ways of thinking. So just want to show you really quickly in the last couple minutes. And this gets back to something else people talked about optimal stimuli for driving neurons. So one of the things people said is, well, maybe we've been thinking about this all wrong. If you're showing an animal or a human lots of pictures, you're not exploring the parameter space very well. So maybe what we should do is we should use a neural network that's been trained on millions of images, that's got a very good mapping of the parameter space of all images, at least the kinds of images people like to look at. We should train that to synthesize images that then optimize the response of the neurons. And effectively, there was two groups that did this almost simultaneously, one Marge Livingston's group, and then the other one Jim DiCarlo's group. And the papers came out within a week of each other. I was a little bummed because we were going to do the same thing in MRI, and we hadn't got it quite working yet. But it's great it works. So they're using a pre-trained deep network. They were training on random textures. They had animals fixate while looking at the images. And what they do is that they had this kind of evolutionary process genetic algorithm where they selected the top images from the prior generation plus 30 new ones that were generated from the network. And they went through 250 generations where they kept optimizing the images to drive the neurons more and more and more. And you ultimately get to the point where I think the next graph, next page has better pictures. You can see, let's see, you get these kind of preferred stimuli. This is like the best images from ImageNet for a given neuron. This is the thing that evolved. So this is something that evolved using that technique, using these textures that were iterated over 250 generations. Here, down here, again, you've got, let's see, these are the stimuli that were shown, and then these are the target stimuli. I can't quite read that. Oh, this is the best that their genetic algorithm produced for the stimuli in terms of what it evolved to. So these are actually things that were evolved images, okay, that it developed. And it's pretty cool. This and the other group that did this were able to find that they could evolve images that did a better job driving a neuron than any picture you'd ever show in the animal from the stimulus set, like from ImageNet or COCO or anywhere else. So there is some kind of potentially optimal visualizable, it's not, again, visualization is probably a bad metric, but there's something that exists in this total parameter space of the network, which is driving that unit, in this case the neural unit, optimally. It doesn't look like a perfect feature or a building block, and it's not something that's just taken from the picture itself. It's some other point in a high dimensional space. But it's a cool different way of trying to do neuroscience, which I think really is the future. That we should be using things where we train on lots and lots of information in the world where we can leverage all the data we have, and we can then take the small number of neural units that we can observe because of the limits, but explore a large amount of the space because we can move in intelligent ways through the big space that we've already predefined. So there's a lot of work where people are trying to do that. I think there's just more here on evolving that. Okay, I want to show you one more thing. I'm going to skip some. I can refer you to the papers if you're curious. I want to show you this because it's fun. Okay, so this is a little task. Some of you may have seen this, but probably most of you haven't, hopefully. So it's an awareness test. Any of you seen this before? Everyone? Only a couple people. Good. Okay, so you're going to follow the team in white and count how many basketball passes they make. So it's really important. Okay, how many passes did the white team make? How many? 15, someone said? 13? Okay, everyone agree with that? How many of you saw the gorilla walk through there, or the bear, or whatever it was? Anyone? Okay, it's a little bit dark, but you can still see it's right there in the middle, and then it bangs its chest, and then it keeps walking. It works fine, actually, if you have the lights down, too. It's just showing you that humans are funny compared to computer vision systems. We are fallible, so our metrics for what should be successful in AI might be different if we're thinking about biological systems, because we attend to the world, we think about particular things in the world. In the case we were attending to the white people making the passes, you're missing a lot of other information. It's never getting encoded. And so computer vision systems don't kind of work that way right now, but ultimately if we want to align them, we have to think about the ways people also fail. And so I think it's an important thing to keep in mind when you're doing this. All right, I'm happy to take questions afterwards. People can come up. All right, thank you.